Question 1. What is the conclusion and outlook of the study as outlined by the authors?
The researchers provide a wide analysis over the genome of orange double haploid type carrot denoted as TH1. Firstly, genome sequencing was done and the complete carrot genome assembly was produced. Then, they characterized the genome assembly and concluded that there are more than 30,000 protein coding genes in carrot genome, which is 20% more genes than human. The researchers also performed an analysis on carrot diversity by comparing wild carrots and cultivated carrots. From the analysis, they found that the cultivated carrots has a slightly lower level of genetic diversity than wild carrots due to inbreeding caused by domestication. This can cause domestication bottleneck. Phylogenetic analysis was also performed by the researchers to provide the insight on evolutionary history of species without using fossil records but genetic fingerprints. The researchers also attempt to find three classes of genes that are responsible for carotenoid accumulation, regulatory genes, and pest and disease-resistant genes. All in all, these findings were said to be important for improving human health and sustaining agricultural production.
Question 2. Briefly explain how the carrot diversity analysis was carried out and what are the corresponding results.
Based on question 2, carrot diversity analysis was carried out by resequencing 35 carrot accessions that represent certain carrot species. And then the carrot species is then filtered and a large number of high quality SNP were identified. 3 studies was done to differentiate the species. First, the phylogenetic and cluster analysis. Second is the nucleotide diversity. And third is the pairwise population differentiation through computation. Okay, as a result, in phylogenetic and cluster analysis, it can be seen in the phylogenetic tree that there are close related of gene between wild and cultivated eastern compared to wild and cultivated western, which is a large difference in their gene. Second, in nucleotide diversity studies, it can be seen that wild carrot have a high genetic diversity compared to cultivated.
Question 3. Based on the phylogenomic analysis, how many plants were compared and what are the highlights of the results?
Question number 3. In this study, the researchers used 13 different plants for phylogenomic analysis. They are as shown in this table. They constructed phylogenetic tree of carrot, which is a member of asterid 2 after we diverged from Roseate 130 million years ago. Ericades 101 MYA, asterid 1 90 MYA. Next, they investigated carrot WGD in its lineage, which they found 2 WGD in carrot, DC alpha and DC beta. They also confirmed that carrot do not share some WGD as horseweed when they diverge into apialis and apicalis clades. They found 7 ancestral protochromosomes dispersed in 9 carrot chromosomes. Synteny of carrot protochromosome A90 correspond to several plants with 2x3 or 3x2 WGD events. These two tables showed that DC alpha overwritten duplicated blocks while DC beta overwritten triplicated blocks. Lastly, ontology analysis of DC alpha event revealed that its gene involved in molecule interaction and protein dimerization activity.
Question 4. Explain briefly how did the authors provide proof of concept on the candidate gene that control the level of carotenoid accumulation.
Question 4. The Y and Y2 gene model only provide the phenotypic differences between white and orange carrot but little on carotenoid accumulation in carrot. Therefore, using two mapping population, Y thus regulates high carotenoid accumulation in yellow and dark orange carrots. Then, using fine mapping analysis, it identifies a 75 kilobyte region on chromosome 5 that harbors the Y gene. However, over 8 genes predicted in the region, only DCAR032551 gene that undergo mutation that segregated with high carotenoid pigmentation. Insertion mutation occur in its second exon that create mutation in yellow and dark orange. Therefore, using the data, resequencing data, the polymorphism detection identified 8 single nucleotide polymorphism in 4 genes, including DCAR032551.